name is Jordan Stelflew. I'm going to be talking today about congenital adrenal hyperplasia. Originally, this disease was discovered in 1865 in patients with intersex anatomy. Research on congenital adrenal hyperplasia did not continue until 1950 when symptoms were found to have come from a break in feedback in cortisol production. The disorder is an endocrine disorder, which means it deals with hormones, specifically corticosteroids and sex hormones. There are 3 different genetic deficiencies that led to the enlargement of the adrenal glands, resulting in this disorder: 11-beta hydroxylase, 21-hydroxylase, and 17-alpha hydroxylase. There are different characteristics for each of the 3 different genes affected, which we'll go over later. This diagram shows the adrenal glands are affected by enlargement. Corticosteroids and sex hormones fluctuate due to this abnormality and deficiency from the genetic disorder. With these levels changing, there are systematic effects and consequences such as hypertension and others that will be further discussed with each genetic deficiency. The disease is an autosomal recessive disorder, which means it can only be inherited if both parents have the gene and it is present on chromosome 6. 11-beta hydroxylase produces excessive androgens, a male sex hormone. In the classic version of this deficiency, there are abnormal developments for both male and female genitalia externally. However, internally, the organs are normal. Sexual characteristics such as hair, deepening voices, and others develop early. The classic version also sees early hypertension. Non-classic versions has normal external genitalia but can result in abnormal hair growth in females and irregular menstruation for women and short stature for men. 21-hydroxylase deficiency is also resultant of excessive androgens with 3 different types: salt wasting, simple virilizing, which are both classic forms, and a non-classic form. The classic versions exhibit early growth spurts, decreased fertility, hair growth in women, male baldness, and irregular menstruation, and the non-classic version can result in no symptoms. 17-alpha hydroxylase results in multiple different hormone imbalances, not strictly limited to androgens. And in this one, hypertension is common, as well as disrupted sexual development, potentially resulting in misassigned genders at birth. Infertility is also a symptom of this specific deficiency. This diagram here shows the 11-beta hydroxylase deficiency and its labels in the genome. It is 2032 bases long, and there are some symbols listed here as well. The 11-beta hydroxylase is the most common deficiency for this disorder. This is a basic inheritance pattern for the disorder, showing affected individuals. In some individuals, as was previously discussed, they can still have offspring. Any of the light blue females or males are afflicted, and as can be seen with some of the individuals, the only way for this to occur is for both parents to have at least one of their genes passed on to be the recessive ones. Genetic testing can be very important for this disorder. For this, one of the most common types of testing done is the deletion and duplication test. Due to different tests being done for different reasons, sometimes testing may not be done until individuals are older or going through puberty or have exhibited other symptoms that may come up later in life. For newborns, it is very important for this test to be, or this type of disorder to be identified, because hypertension and salt wasting in newborns can be fatal. The testing is not mandatory, however, for safety, it likely should be highly recommended at the very least. These are the references that I used for this video and research on the topic. Thank you for listening.